Hello friends, this is the third lecture of our endocrine pancreas series. How increased blood glucose stimulates the insulin secretion. Now that is the biphasic response. It is a biphasic insulin response. So whenever there is an increased blood glucose level, it will increase the insulin secretion. It will increase the insulin response two times. Right, it is known as biphasic insulin response. Let me show you on a graph so you can easily understand that thing. Over the y-axis it is the plasma insulin, over the x-axis it is a time. So you can see that whenever there is an increased blood glucose, there is a one peak and there is another peak, right? So insulin secretion will be increased two times whenever there is an increased blood glucose level. So first time it increase within 3 to 5 minutes, and this insulin secretion is about 10 times than the normal level or resting level. And the reason of that release is basically release of preformed insulin. So let me show you some of the figures that you need to remember. Fasting insulin level, that is 0 to 70 micro unit per ml, and the actual secretion per day is 40 unit per day. This is about the first phase or first time whenever insulin get released. Now let's discuss about the second time. The second time insulin secretion will be increased after 5 minutes of the actual glucose in the increase in blood glucose. Now that secretion is higher than the first phase, and the reason behind that is that in second time increased insulin secretion is done by increasing the synthesis and release of the new insulin. Now the other factor that control the insulin secretion. Now the other factor, the first one is the amino acid. Amino acid has the same effect as the glucose has. So what they do, they increase insulin secretion. So whenever there is more amount of amino acid in your diet, it will increase blood glucose, it will increase insulin level, same as blood glucose. And the most potent amino acid are the arginine and lysine. Remember these two, that are frequently asked in MCQ, arginine and lysine. Now if you give glucose and amino acid simultaneously, that amino acid is going to potentiate the glucose effect on the insulin secretion. It is more or less like a synergistic action of the amino acid over the glucose. Now the effect of GI hormone, gastrointestinal hormone, in insulin secretion. Now as far as the GI hormone is concerned, GI hormone is increase in the insulin secretion. First let me enumerate some of the gastrointestinal hormone. Gastrin, then the secretin, then CCK. You know the meaning of CCK, right? It is cholecystokinin. Then the GLP-1. Now what is GLP-1? It is glucagon like peptide 1, and the next one is GIP. Now GIP means glucose dependent insulinotropic peptide. Previously it is also known as gastric inhibitory peptide. Now apart from these five, the last two, GLP-1 and GIP, are the most potent in increasing secretion of the insulin. Now they are known as incretins. So whenever you heard the term incretins, it has something related with the insulin. They are basically increase in the insulin secretion, and the GLP-1 and GIP are the incretins, right? So several drugs have been developed to mimic or enhance the action of the incretins, this GLP-1 and GIP, and they are basically used in treatment of diabetes mellitus. Got it? Now up from this very slide, there are chances that there is one comment might actually ask why oral glucose release greater amount of insulin than intravenous administration of the glucose. The answer, you should write it down like that. Oral glucose increase release of the gastrointestinal hormone, and you have to enumerate all the GI hormones: gastrin, CCK, secretin, GLP-1 and GIP, right? Now this hormone will potentiate the release of insulin. So when you administer the insulin, when you administer the glucose IV or intravenously, that intravenous glucose administration do not release the gastrointestinal hormone. So they are not able to potentiate the insulin release, right? So that's why the oral glucose release greater amount of insulin than intravenous administration. Now effect of ANS over the insulin secretion. Now pancreatic islets are richly innervated with ANS, autonomic nervous system. And this autonomic nervous system has a two division. One is the parasympathetic, another one is the sympathetic. So the parasympathetic division is actually stimulate the insulin secretion, and the remaining one as the sympathetic stimulation, it is going to decrease the insulin secretion, while on the other hand it will increase the glucagon secretion, right? Now there is one question that sympathetic stimulation, this sympathetic supply has a two type of receptor. One is the beta receptor, another one is the alpha receptor, right? Now beta receptor is going to actually stimulate the insulin secretion, and alpha adrenergic receptor is going to inhibit the insulin secretion, right? I give you homework in our first lecture of our endocrine pancreas series, if you remember, right? Beta adrenergic stimulation will increase the insulin secretion, and alpha adrenergic stimulation will decrease the insulin secretion. But more potentiate the effect, a more pronounced effect is the inhibitory effect. So in summation, sympathetic stimulation will ultimately decrease the insulin secretion. That is the answer of your homework that I given you in the first lecture of our endocrine pancreas series, right? Now the role of insulin in switching the source of the energy, it is very important, and it is very beautifully given over here. Now role of insulin in switching the source of energy. So you can see whenever there is an increase in insulin secretion, it will increase the utilization of the carbohydrate and ultimately decrease the utilization of fat. Got it? So what will happen whenever there is a decrease in insulin secretion or lack of the insulin secretion? Lack of the insulin secretion is ultimately leads to decrease utilization of the carbohydrate, and in the reverse manner it will increase the utilization of the fat. So you can see only presence or absence of the insulin is actually switching the source of energy, right? In the same fashion, whenever there is a glucose concentration is low, when the blood glucose concentration is low, is basically decrease the insulin secretion, and ultimately fat is used for the energy. So when glucose is low, fat is used for energy. In the opposite manner, when the glucose concentration is high, it will ultimately leads to increase insulin secretion, and carbohydrate is used as a source of energy. Alright, this is how the insulin will switch the source of the energy that we used. This is all about the insulin. Now this phenomenon of switching is true for all but except brain. Alright, now this is all about the insulin. Now next hormone that we are going to discuss is the glucagon. Now as you guys see that glucagon is secreted by alpha cell of the pancreas, and it is also a polypeptide hormone. So it has an amino acid sequence, it has a 29 amino acid in its molecule. So its action is basically increase blood glucose level. So that action is exactly opposite of the insulin. Insulin is going to decrease the blood glucose level, while on the other hand glucagon will increase the blood glucose level. That's why glucagon is also known as the hyperglycemic hormone. You know the other hypoglycemic hormone that I mentioned in our growth hormone series? Hyperglycemic hormone are starting with the G, glucagon. Second G is the growth hormone. Third one is the glucocorticoids. Alright, and the catecholamines obviously. Now how the glucagon will act? Effect of glucagon. So whenever there is an increase in blood glucose level, effect of glucagon is it increase the blood glucose level. How it will increase the blood glucose level? First thing first, it increase availability of the glucose. So basically it break down the liver glycogen and it break the polymerization of the glucose, that is glycogen, and increase the availability. This is the one source. Second source, it increase the formation of new glucose. It will increase gluconeogenesis. That's how glucagon will increase blood glucose level. So how glucagon will increase blood glucose level? First is it increase breakdown of the glycogen, that is glycogenolysis. Another one, it increase gluconeogenesis, right? But how the glucagon causes glycogenolysis? The answer resides is that in the action of the glucagon. Glucagon will use cyclic AMP second messenger system. Let me show you a diagram. You guys already know about the cyclic AMP or the cyclic AMP second messenger system. You guys already know all the sequence. Whenever the ligand bind with the extracellular domain, it will causes conformational changes inside the intracellular domain. It will bind with the G protein, and inside this G protein there are three subunits, alpha, beta and gamma. Alpha subunit get detached and it will bind with a enzyme. That enzyme is adenylyl cyclase. Once this adenylyl cyclase is activated, it convert the ATP into cyclic AMP. This cyclic AMP will causes phosphorylation of the protein kinase and make activated protein kinase, and then it will causes the changes inside the cell, right? So this is the action of the glucagon. Now how the glycogenolysis will occur? So glucagon will first activate the adenylyl cyclase in the hepatic cell membrane, then it will increase the formation of cyclic AMP. Now once the cyclic AMP will form, it activate the protein kinase regulator protein, that is protein kinase A, right? It is activate the protein kinase A. Now this activated protein kinase A will activate another protein kinase. That protein kinase name, remember the name, it is phosphorylase B kinase. Phosphorylase B kinase. Now it converts the phosphorylase B into phosphorylase A. Remember that thing, phosphorylase B kinase is an enzyme that converts phosphorylase B into phosphorylase A, and our main thing, that is phosphorylase A. So this phosphorylase A ultimately increase the degradation of glycogen into glucose 1-phosphate. It will cut down the glycogen into fragments, that is glucose 1-phosphate. Then glucose 1-phosphate, there is a phosphate radical attached to it, then again it is dephosphorylated and glucose is released from the liver cell. This is how the glycogenolysis will occur. Let me repeat one more time. First thing first, glucagon will act via cyclic AMP second messenger system. Ultimately it will activate the protein kinase A. This protein kinase A will activate the enzyme phosphorylase B kinase. It will convert the phosphorylase B into phosphorylase A, and this enzyme phosphorylase A will causes the breakdown or cut the glycogen into pieces, that is the glucose 1-phosphate. Now still glucose has a 1-phosphate attached to it. This phosphate should be removed, then and then glucose will be diffused outside the hepatocyte, right? So it is dephosphorylated and glucose will be released from the liver cell. This is all about how glycogenolysis will occur. Now how gluconeogenesis will done? Let's discuss that thing. How glucagon will leads to gluconeogenesis. These are the steps of the gluconeogenesis. Now glucagon is actually increase the rate of the gluconeogenesis by two steps. First step is it increase the rate of amino acid uptake by the liver. Got it? First thing the glucagon do, it increase the uptake of amino acid by the liver cell. Once the liver cell has enough amount of amino acid, then glucagon will stimulate the conversion of this amino acid into the glucose. That thing is known as gluconeogenesis, right? Formation of the glucose from non-carbohydrate source. In this case, the non-carbohydrate source is amino acid. Now, how it stimulate or convert amino acid into glucose? It by stimulating the multiple steps that involved in this gluconeogenesis pathway. This is the pathway starting from the pyruvate to this entire sequence, and glucose will be made. So glycogen will ultimately activate the enzyme that involved in the gluconeogenesis. The most important is basically enzyme that convert pyruvate to phosphoryl pyruvate or phosphopyruvate, or this phosphoenol pyruvate, right? This is the main step of making gluconeogenesis by conversion of pyruvate into phosphoenol pyruvate. Got it? Now, which enzyme is basically responsible for this conversion, pyruvate into phosphoenol pyruvate? This thing is actually covered in biochemistry, but let me remind you that the enzyme responsible for this conversion, pyruvate into phosphoenol pyruvate, first is the pyruvate is converted into the oxaloacetate, and that oxaloacetate is ultimately converted into phosphoenol pyruvate. So the enzyme that convert pyruvate into oxaloacetate, it is known as pyruvate carboxylase, and the enzyme that convert oxaloacetate into PP or the phosphoenol pyruvate, it is known as phosphoenol pyruvate carboxykinase. This thing might be confusing, so let me show you one slide picture like this. So this pyruvate is ultimately going to converted into PEP, that is phosphoenol pyruvate. So to order to make phosphoenol pyruvate from pyruvate, this pyruvate is first converted into oxaloacetate, and oxaloacetate is ultimately make phosphoenol pyruvate. So the enzyme responsible for this conversion, pyruvate into oxaloacetate, is the pyruvate carboxylase, and the enzyme responsible for conversion of this oxaloacetate to phosphoenol pyruvate, it is phosphoenol pyruvate carboxykinase. So if you do not remember this sequence, that is okay. Average student should be remember up to this level. The glucagon will stimulate the gluconeogenesis by stimulating the enzyme that convert pyruvate into phosphoenol pyruvate. But the brilliant student will also remember this sequence also: pyruvate to oxaloacetate, oxaloacetate to PEP. Right? This is the rate limiting step. This one, conversion of pyruvate into phosphoenol pyruvate, is the rate limiting step of the gluconeogenesis. Now these are the effect of glucagon once it is present in the normal level. What happens when the glucagon will be present in the higher amount, more than it is actually found in our tissue, found in our blood, right? So the other effect of the glucagon, first effect is that it enhances the strength of the heart. It increases the strength of the heart. Second effect, it increases the blood flow in some tissue, especially in the kidney. Third thing that glucagon do is enhance the bile secretion. And the last one, it inhibits the gastric acid secretion. Now all these four effects are not present whenever the glucagon secretion is in the normal limit. It is only found that the maximally present inside the blood, right, when the glucagon level is raised more than normal, then and then the effect of glucagon over the heart, blood flow, secretion of the bile and gastric acid secretion will be seen. Alright. Now the regulation of the glucagon secretion. So first thing that is responsible or take part in the regulation of the glucagon is the blood glucose level, obviously, right? So whenever there is a decreased blood glucose level, it will increase glucagon secretion. Whenever there is increased blood glucose level, it will decrease glucagon secretion. So first enumerate the different factors. One is the plasma glucose, blood glucose, then the plasma amino acid, then the gastrointestinal hormone, and the ANS, autonomic nervous system. So the effect of blood glucose is like this: whenever there is decreased blood glucose level, is ultimately increase the glucagon secretion. Whenever there is increased blood glucose level, ultimately decrease glucagon secretion. Got it? In the plasma amino acid, whenever there is increased plasma amino acid, it will increase glucagon secretion, and glucagon increase the conversion of amino acid into the glucose. Now effect of GI hormone is that cholecystokinin, gastrin and GIP is ultimately increase the glucagon secretion, and somatostatin will decrease the glucagon secretion. And as far as the ANS is concerned, sympathetic stimulation is basically stimulate the glucagon secretion. Got it? This is all about our today's lecture. In next lecture we are going to learn about the somatostatin and the diabetes mellitus. Thank you.